No, I was just about to send an announcement about office hours for 420.
Oh yeah.
For tomorrow's discussion.
Yes.
I mean, in church. That's your week.
Yes,
Yeah.
right. The group assignments went out. So. I should have been asked before, but I just used them.
Oh, you already sent them?
I've sent
so
them.
they're already
It's already issued.
written in stone?
Yes,
Okay.
already.
Okay.
Yeah,
Okay.
I
So
did it.
yeah, they just, in the past, we have reviewed them. But I also looked at them today.
Right, yeah, I think the collab presumably relatively new as well.
Well, the whole discussion is only from last year.
Right, yes.
We made it last year.
Questions seem still pertinent.
Yeah. Oh, hey there. How are you?
Are you gonna bring the dog to the dogs?
No, I do sometimes, but he's a little overexcitable.
Yeah.
It's an exciting talk. Yeah.
Our dog is fine.
Yes, I was just
Yeah.
saying,
Our
so
dog
you're
is
fine. It's
fine.
not the
He just
little
goes
one.
flat.
Yeah.
Yeah, he's totally cool.
The little
Sometimes our
ones
dog
are
has a
big.
laugh, so.
Yeah.
It's
Yeah.
really
Yeah.
a
Yeah.
dog.
I know.
He is, yes. It was boring earlier. Yeah. It's today. Yeah, just keep working. Well, that's why you're late. That's what she said, you were late. Coming here next week. That's why he's like, call that Boston Hotel, and it's like, oh, the Renaissance. Yeah.
Yes.
Yeah.
Yeah.
I always check my car with.
Okay.
Yeah.
And then I said, that's a 5 minute walk.
Yeah, it's a pretty experience. So okay, there they are.
Let's go. Down here. So then I pull up and I realize,
But we can.
this is like, oh, I better been at this house.
So she went to like
That reminded me of Rebecca Sachs. She, you never told us story, that story. She, I guess she showed up with. All friends was there. I really understand this story, but she
She's
was
like, what
supposed
the hell just happened?
to.
I know there. Was it not enough? Yes. Yes, it is.
You know, I mean, it's like, wait. It's like 2 hours away. Yeah, so she was planning it.
Yeah.
No, you guys didn't fly.
I'm gonna go to
I was signed up for it all last year and I didn't make it a single time.
yeah.
But it's good to know what they're reading.
Yeah.
But I got a Slack invite and I'm like, I don't know if I'm gonna join that Slack.
If you have any interest, also this is random. I just got invited to an optimal transport. I'm not gonna go.
Hello and welcome. Hi guys. Hi everyone. Welcome to the first colloquium of the Simon Center for this term. And it's my pleasure to introduce Dr. Todd Constable, who is coming from Yale. So Todd's Canadian. He got his undergraduate and master's degrees in Manitoba in physics, and then a degree in biomedical physics from U of T, University of Toronto. And then that was in 1990, and then he moved to Yale Medical School, and he's been there pretty much since then, so over 30 years. And since 2001, he has been directing the Magnetic Resonance Center, Research Center at Yale as well. And he now holds the title of the Elizabeth Mears and House Jameson Professor of Radiology and Biomedical Engineering. And I don't want to take up too much time, but I just think there's two things that are really quite amazing about Todd. One is that he's been wildly productive. He has almost 400 papers in peer-reviewed journals and over 52,000 citations. That's a lot. The second is really the diversity of the questions that he's tackled and the approaches that he's been using in his work. His main, he kind of has two lines of work. One, probably the bigger one, is trying to use magnetic resonance imaging to understand the functional organization and connectivity of the human brain and relate neural features to behavior in neurotypical individuals and diverse populations with brain disorders and psychiatric conditions. But then he has, and most of you may have heard some of that work where Todd's group reported that you can identify individuals based on patterns of whole brain functional correlation patterns, functional correlation data, so-called brain fingerprinting from a few years ago. And then the second line of work is using his background in physics to build more accessible MRI devices using low field imaging to eventually hopefully have these devices in doctors' offices and be able to have them accessible to populations that haven't had access to that technology historically. So Todd will tell us about brain and behavior today, so welcome, Todd.
Thank you very much, Ev. It's great to be here, and what a great day I've had. I've had some really good conversations with people, as I anticipated, and so that's really nice. So thank you, everybody, for having me and hosting me. I'm going to kind of do as Ev kind of alluded to, I hate to say I'm a methods person, but I guess I'm a methods person. So I'm going to talk about a bunch of different methods that we've been playing with in terms of understanding the brain through looking at brain behavior relationships. And I think there's some cool things that we can learn, and I'm excited about this whole field. And so I'll kind of go through, I think there's 3 different phases or something. I got too many slides in here. Feel free to interrupt me at any point. I'm happy to discuss things, but I can also kind of barrel through and see if we can get through all the slides. I want to talk about brain behavior models in general. How do we do it? What the best data is for that, and what can we learn from these brain behavior models? And then I'm going to talk about some more recent work, which is, how do we get more from these models? How do we use priors? So, for example, define the networks of interest. And then how can these models tell us something about the behavioral measures that we're using to build the models, and in turn those behavioral measures tell us something about the brain. So it's kind of a feed-forward feedback thing that I want to emphasize. And I think from some of the conversations I've had, some of that stuff might be useful for people here. And then, as Ev pointed out, I'm very interested in clinical applications. We're collecting a big transdiagnostic data set of people with a variety of psychiatric illnesses, and we're trying to look at common themes and common circuit dysfunctions in maybe identifying groups or subgroups of people in that sense. And so I'll talk a little bit about that in terms of applications. If you want to know about prediction, John Gabrielli here has written kind of a great paper on this. This is kind of, we read this a lot when we were starting out. So you have the source right here, and that's a great paper to look at. And there's been obviously lots of work since, but that's kind of where we started. So I'm going to talk about, when I talk about these brain behavior models, I'm talking about linking the brain, so either structural information or functional information or both. I'm mostly going to talk about functional information. Linking the brain to behavior, and that behavior is typically scores from standardized tests. So these are reaction times, accuracy, or any sort of quantitative score that you can use. There's a lot of self-report information out there and clinical measures, and that's ultimately what we're really interested in. Or if you're going to put this in a clinical context, most of those measures are self-report, and so we'll talk about that. There's a side issue of state versus trait, so I'm mostly going to focus on traits, trying to predict traits through these models, but they are sensitive. The same models are sensitive to state changes or state manipulations. And Monica Rosenberg from Yale, who did her Ph.D. with Marvin Chun and worked with us a lot, she's got a lot of papers on state-dependent kind of attention information. And so you can do both. I'm mostly going to focus on trait. So why do we build such models? So the goal is not really to predict behavior. I sometimes get asked, why don't you give them a pen and paper and do the behavioral test, right? It's a lot cheaper than putting them in an MRI scanner. So the goal is not necessarily to predict behavior. The goal is to reveal the underlying brain organization or the circuits that support test performance. And I emphasize their test performance. So it's not necessarily the circuits involved in executing that behavior, but it's the circuits that are preferentially involved as a function of how well you do that, perform on that test. And so there could be lots of circuits involved that aren't sensitive to performance. And then fundamentally the goal is a clinical marker for brain disease or disorder. And why do we do predictive models? Simple association studies, as you saw from the Merrick paper in 2022 in Nature, simple association studies are weak. They're generally not reproducible. That was the message of that paper. And if you can take an association and build a predictive model from it and get some predictive power, then that makes you believe the association. And so then I would say that's a more meaningful association. And actually, Monica and Emily, who are both grad students at Yale and now are both faculty, one at Northwestern and one at Dartmouth, had a nice kind of rebuttal to the Merrick Nature paper, which is that if you want robust brain behavior relationships, you need to build these sort of predictive models. So how do we do this? What are we talking about? So we get something like 100 subjects. It depends what you're modeling. Maybe if it's attention, you could do this with 40 subjects. But 100 is nice, 1,000 is better. You get a bunch of subjects, you build a connectivity matrix, and you have some sort of behavioral measure, a test score or something. And what we're doing is we're finding the relationship through correlation generally between this connectivity matrix and this behavioral measure. So this will identify the circuits supporting performance. And so what we're doing is finding the edges or the connections that correlate across subjects with this behavioral performance. And so just if people here aren't doing bold imaging, these are fMRI experiments where we collect brain volumes, let's say once every second or less. And we may collect 300 volumes or 2,400 volumes, something like that. And we correlate, we impose an atlas on the brain, and then we take the time courses from that region through time, and we correlate that with another region. And the whole basis of functional connectivity in fMRI is that if these regions are correlated, they tend to be functionally connected, or we assume that they're functionally connected. And that gives us a matrix. The dimensions here are the dimensions of the atlas. So if we have a 268 node atlas, this is a 268 by 268 node matrix, and it's symmetric, okay? There's no directional information here. fMRI in general is too slow to give us directional information, so the connection between A and B is the same as the connection between B and A. So we really only need to look at one half of this matrix on one side of the diagonal. Is that clear to everybody? So each element of this matrix is an intersection between two nodes, and that's how we fill this matrix here. So the whole talk is basically on this connectivity matrix. This is what it looks like for a 268 matrix. It's actually pretty dense. And this is actually, I guess, fundamentally, this is what I think is the cool thing about this. There are 35,000 connections here in this 268 by 268 matrix. If we use something like the Glasser atlas, which is a 360 node atlas, that's 70,000 connections. What I find so fascinating is that these connections, I've referred to this already in our Nature Neuro 2015 paper, we showed that we can identify you by these connections, by your particular pattern. So there's like a brain fingerprint. But these connections tell us something about the individual. So these tell us something about your traits. And I think we're really still, this was, let's say 2015, or people were doing some of this stuff before that even, but we're still just at the beginning of learning how to extract the information. So from these 35,000 or 70,000 connections, we're still learning how to read that code, what's in that code, and what can that tell us about the brain. And I'm quite interested in that that question. So that's the human connectome. When you hear about the connectome, well, this is one form of the connectome. This is the functional connectome. There's also a structural connectome if you're looking at the diffusion tensor imaging and look at the structural connections between nodes. But I'm going to focus on the functional connectome. So how do we build our models? Let's get right to the models. We stack up a bunch of matrices from n subjects. And we have some behavioral measures, so each subject contributes a behavioral measure. And what we do is we correlate those. So we correlate this vector, this behavioral vector here, with every element in these matrices. And so here we have a matrix of correlation coefficients, which is, when the score goes up, which connections go up, and when the score goes down, which connections go down. And we get a positive tail and a negative tail. We can threshold those. And basically these dots here represent intersections, represent pairs of nodes that are changing as a function of score, so either going up or down with score. So that's an association. So we've established an association between edges, connections that vary with behavior. So this yields a network, and they either vary positively or negatively. This is simply an association study, which we know is not reliable. So the next thing we do is we're going to build a predictive model from this and see, can we bring in a new subject and predict what their attention score should be, or their D prime score for whatever this measure is here. So let's look at this positive tail. We can just add up all of the connection strengths from those edges, and we get a network score, and we can plot D prime for each individual subject. So each of these dots here is a subject, and each of these subjects has a network score for the sum of these edges in their connectivity matrix. And we can then fit a line to this, y equals mx plus b. That's the sophisticated math we're applying here. So we fit a model to that. And then we can bring in a new subject, and we can say, given this kind of mask here, if we calculate what their network score is, can we predict their behavioral score? And we can do that for many left out subjects, and we get a plot like this, which gives us some sense of our predictive power, how well we can do with this. And we can measure that model performance. The best measure is like a mean square error on the fit. Typically people report R squared, and actually I'm going to report R here in a lot of these things. But you want to measure mean square error between the predicted and the observed value. You can do this with a negative tail. Basically, you can do it with both. In a GLM sense, the results don't change very much with either the positive or negative. And it turns out that the masks don't change much. The nodes that are contributing the positive edges are also contributing the negatively correlated edges as well. But that's the model, basically. What's the fit between network score and behavior? And then this is really the validation here. If this model performs well, let's say we have an R between 0.3 and say up to 0.6, I'd say that's a pretty good model. So I'm going to focus on that. What does that yield? So if you threshold this matrix, and plot it on a circle plot like this, the front of the brain is here, the back of the brain is here. This is the right hemisphere, this is the left hemisphere. So cerebellum's back here, frontal cortex is there. And these are all the nodes in that, in that hemisphere. And these are all the nodes in the left hemisphere, and these are the connections that are revealed here in the positive tail. So this actually is a plot from an attention task. So you can see a lot of stuff gets activated, and this is kind of, immediately you want to say, well, that just doesn't make any sense. Attention's prefrontal cortex, and look, you got everything here. This is actually to me, I would argue back that, well, this is probably a more realistic depiction of how the brain works, right? The brain's complicated. It's not one region doing one thing. Everything's connected, and there's a lot of support for doing things. And when you look at the numbers here, so even if we have 1,400 edges, that's like 4% of the available connections. And so the fact that it's complicated is kind of our problem, not the data problem. So this is pretty far away then from region A does this. This is like, what is the system supporting behavior on this task? And so we have to think about it a little bit differently. Typically we want to, our natural question is, well, what's driving, performance? And sadly, I don't think it's like one region that's driving performance. That's not to say that you can't lesion somebody and destroy their performance. but clearly there's a powerful network that serves brain function. And some of the best predictive models we have in the field come from this sort of thing, where we're using connectivity matrix and behavior to generate these predictive models. So I'm going to argue here that the brain is complex, and you should be comfortable with this sort of data. It doesn't have to be resting state data. I had a conversation with earlier today that they weren't getting good predictive models with resting state data. Resting state is kind of the worst condition to collect connectivity data in. It's certainly easiest. The patient doesn't have, or the subject doesn't have to engage in any task or anything. But it's actually, because it's so uncontrolled, there's a lot of additional variance, I think, introduced by the resting state, which you can eliminate by synchronizing everybody's brain with a task. And I thought, early on, that, well, if we want to measure attention, if we give somebody an attention task, maybe that's a better way to tease out differences in attention, right? And you think about a cardiac stress test. If you look at a patient with heart disease, if you look at them at rest, their heart may look normal. But if you make them exercise, you give them adenosine, or you put them on a stationary bike, you'll see who's got a healthy heart and who doesn't. And so, if you take the stress test analogy, maybe giving an attention task would help us build a better attention model. So we want to collect data. These are continuous performance tasks, so we're not really looking at blocks of on-off activations or even event-related. These are like continuous performance tasks, and we think of them as changing the brain's state by giving a continuous performance task. And so we get a slightly different connectivity matrix for each run. It's slightly different in the sense that some regions are stronger, some regions are less. There is an activation effect for some of the few regions that activate in a given task. But this is from the HCP data and Emily's work showing how well we could ID somebody under different conditions. And in general, you can still identify people by their brain, whether they're doing rest or language task or working memory task, as these different boxes show. So the patterns change slightly, but you can still identify people. They don't change dramatically. And I think of this as almost like how your brain changes slightly when you do a task. You get a 2% activation change, but your brain still looks like your brain. And so the variance between our argument for doing a task is that the variance is reduced by synchronizing subjects. So you could then have a suite of continuous performance tasks. And I guess, further on the cardiac analogy, if you could tweak the system all along the attention network and then along the memory network and some of these different dimensions, maybe you could span the space of the relevant cognitive functions, and putting all these connectivity matrices into your model might give you better prediction. In general, we just append these when we're doing this, or we just use data from like a movie run or something like that. But here's some data from the HCP. If we took all the HCP tasks, so the resting, this is the percent variance explained. It's actually pretty low here, but in the resting runs, rest 1 and 2, the percent variance explained is very low. This is in predicting fluid intelligence, I think. But like the gambling task, for example, did the best job of prediction. And then similarly, if you look at the PNC data, that's from the Philadelphia neuroimaging cohort, again, the working memory task here does better than an emotional memory task, and then better than rest. And so we've done a bunch of investigations into trying to figure out how this, what's the mechanism here? Is it task effect? Is it intrinsic connectivity? It's a little bit of everything. So there is a task effect, which accounts for maybe 30% of the variance. Intrinsic connectivity accounts for about 30% of the variance, and I forget what the third thing is, but the interaction between them, I guess, is the other 30%. And then Emily here subsequently wrote a paper, is it time to put rest to rest? And the implication there is that resting state is not a good condition in which to collect data. So we do a suite of tasks. We have a memory task, an attention task, we have a movie condition, and we're trying to kind of tweak the brain on these different dimensions in order to build better models. And I'll talk a little bit more about that in a second. So we have 16 behavioral measures in this data set that we're collecting in a transdiagnostic sample. And actually there's a lot of subscores here, but we can boil them down to these 16 measures, and these are kind of standardized tests. And what we want to do is build models for all of these. But again, coming back to the coolness of this problem, information on all of these traits, these 16 measures here, is in this same connectome, right? I mean, so we can build models from this same data for any trait that we want to look at, and I think that's kind of cool. So here's the R values for predicted versus observed for these different 16 measures. You can see some of them are really poor, like this one here, 0.01. That's not really working. But we can also, this is for resting state, we can also do this under our different task conditions. So here's card guessing, emotional eyes, grad CPT, movie, NBAC, and stop signal. And the red boxes are the tasks that gave us the best predictive models for that behavior. So the columns are the behaviors, and the rows are the different continuous performance tasks that we did in the magnet. So the first take here is that movies is pretty good. We got 5 wins there for movies. And actually, surprisingly, the emotional eyes task is actually pretty good. Another 5 for that. So 10 of the best predictive scores came from those 2 conditions. And so we're still, it turns out it's not the cardiac analogy that I was giving you. For example, the attention measures here are not best in the grad CPT task. And so I think it's more along the lines of the synchronization. The card guessing task is the worst task for building these predictive models. Resting state's also not very good. But the emotional eyes and the movie condition were the best. And we still don't really know why these are different. If it was just synchronization, one of these tasks would win, right? Because it would be removing all the variance from everybody, and then we should be able to do good modeling for all of the things. So it's not just synchronization. It's not really the cardiac analogy. So we still need to sort out what the different components are, and I'm going to come back to this in a little bit. But again, I'm emphasizing that we can do all these measures from the same data set, and one of the things that I've said to several people here that I'm going to gloss over here, but I just have to throw it out there and make everybody think about this problem. So we're fine with looking at connectivity changes here. We get a different connectivity matrix for different task conditions, and we're fine with like using a fixed atlas. I would argue that that's actually wrong, and that the atlas, the functional atlas here, the nodes reconfigured with task, and I've got a bunch of papers on that now. And other people have shown in other situations that there's lots of functional reorganization as you change conditions, and most people are comfortable with that, but most people are also comfortable with using a fixed atlas here. And we've started doing the reverse of if you go backwards and try to derive the atlas in the reverse process, what do you get? Or do it jointly, so build the atlas and the model at the same time, and you get different atlases. And so I think that we're going to have to think about flexible brain organization within a fixed infrastructure, and that's something that I get a lot of pushback on reviewers from, but I think I'm right. And it's the fact that for 150 years, all of neuroscience had was microscopes and cytoarchitecture. And it's only recently, so they throw back at me, well, we have 150 years of neuroscience supporting this. And I go, yeah, but now you're looking at anatomy to learn something about function. Now we can look at function more directly, and now you want to ignore the function. So I don't know. That's the arguments I have with reviewers, and I always lose because they have the power, right? So, but someday I think I'm trying to get everybody on board, and as you start to think about this, maybe we'll get somewhere. So where are we? So we get these complex patterns of connectivity. We have one of the things that comes out of these behavioral measures is some things are easier to model than others. By that I mean they yield better predictive power. And what I think when I think about interpreting that, what does that mean? That means that the test that we have has a more pure brain correlate, okay? So like if there's clearly some clear brain correlates that relate to performance on the task, we can find those. If it turns out that you're not getting good predictive power, there might not be a clear correlate, meaning maybe there are different strategies people can engage, different ways they engage their brain to do the same task, and so then you can't get a good model. But I think this is a very interesting thing to look at in more detail. So some test measures, and ultimately when we want to go to symptom scores, don't have clear correlates. And one of the examples I give is we've worked with Dylan Gee at Yale quite a bit on anxiety, and she's got a bunch of different anxiety measures in different populations and things like that. And we've tried with sample sizes of 150 subjects and things like that, and we can't predict anything with these anxiety scores. And yet we can take almost any data set that has attention scores and we can predict attention. So there's something very different going on with these different things. And one of the things is what we want to do ultimately, though, is build these models in a population and then apply them to patients or individuals and see how these rank on a norm distribution. So we're trying to build up a sample where we can get distributions here, and so we can see who's within and who's outside of that distribution, and try to sort that out. So I'm just going to emphasize why this figure, that with these 16 different measures, we get 16 different networks supporting those measures, and so that's kind of what we get from the brain behavior modeling. Yeah. Yeah. Yeah, you could do both. You could, so this boils down to a, we sum up all the edges and we get one number, right? You could have two numbers where the second number is the magnitude of activation, for example. You could bring in all kinds of other things. And at the end of the talk, I'm going to talk about bringing in other things. But yeah, by all means, you could do that. So how do we get more from the data? I'm going to kind of keep motoring on here. So far, we've used this matrix as our one item in our data-driven approach to modeling. What I'm interested in is knowing, like, we know something about the brain, so let's a priori, break up the brain into, say, 6 networks and look at the contribution of each network. So we're going to skip this step here in the middle, which is to identify the networks. We'll just identify the networks independently. And we did that by going to Neurosynth. So we went to Neurosynth and we took the 6 RDoC, which I'll explain later, cognitive constructs, which are attention, perception, declarative memory. We took those 6 constructs, and using Neurosynth, we searched, what are the nodes associated with attention and perception from thousands of studies. And we selected those nodes and made a network of those. And then we're going to kind of do the same thing, but now we're going to model this again, performance on a task versus these subnetworks. And we're going to see what's the contribution of each of these networks to that behavior. So we end up getting a weight, these, the slope of the line, let's say, how much each of these systems is associated with performance on the behavioral test. And this is, I think, the first time, I think this is the first time where we've been able to take a behavioral test that's been used for 100 years in behavioral testing and, look and see, well, what is it really, what systems is it really relying on? And so here we can, if we look at working memory, so we're looking at the working memory network and its contribution to performance in all these different 16 tasks, and we can kind of rank that. And this is after many randomizations and things. That's what the error bars are. But this is saying, like, verbal fluency. Performance in this verbal fluency task is strongly weighted by this working memory network. And there's very little, in matrix reasoning, there's very little working memory. That's what this is saying. We can also turn that around a little bit. So we can do this for all the different constructs. So that's the working memory, there's the attention, and the y-axis is changing here. There's the language network. This is a poor language network, and I'm happy to, or I freely admit that these so the point here was to demonstrate the method. There's a lot of refinement that needs to go into how we define each of these networks, and that's an open question right now. And actually, I've talked with Ev about using her language network, which is probably much better than the network we get from Neurosynth, to use as input here. Another way to look at this is if we take a given behavioral task, let's say verbal learning delay, we can put them, the 6 constructs there, and we look at the weighting on each of those constructs and see, is it mostly driven by perception, working memory, language, whatever. And so you can generate this sort of plot for each of the for each of the behavioral tests. So there'd be 16 of these circle plots here. So I think that's kind of cool, and I don't know that I've seen a lot of this in the literature on these standardized behavioral tests. So we have assumptions as to what the behavioral tests are measuring, but now we can actually get data and say, well, to what extent are these systems contributing to performance on those behavioral tests? So if you develop a new behavioral test, one of the things you might do is run it through something like this and see what is it really, what systems is it really relying on. And so there's kind of a feed-forward feedback loop that you could get into there of tweaking your test and seeing does it emphasize more attention or more memory or more language. So I do think that that's potentially useful in this whole, developing behavioral tests and learning more about the brain systems that they rely on. So there's another way to look at this feed-forward feedback, and this is, we basically have here, we have 6 network weights, so that's what, let's say these are the network weights, and we have 16 behavioral measures. We threw this into like a hierarchical clustering algorithm to see which of these tests, the behavioral tests, activate the brain in the same sort of pattern. So by activate the brain, I mean have a M1 to M6 vector in the same sort of shape. And so we can do hierarchical clustering on that and find behavioral measures that rely on the same networks. And when we do that, here's our 16 measures kind of reorganized now along the x-axis. So the numbers on the x-axis are the behavioral numbers in the table here. So behavioral tests 1, 13, 2, 10, and 16 seem to rely on those 6 cognitive construct networks in the same way across subjects. And so what can we do with that? Well, so if we take those, there's 1, 2, 3, 4, 5. If we take those 5 behavioral tests, and we just average the scores, and we redo our connectome-based modeling, we actually do a lot better on our predictive modeling, meaning that we're somehow zeroing in on a behavioral score that's more consistent for each subject. And so if we look here, this is the predictive power of the individual tests. So behavioral test 1, 13, 2, 10, 16. And then this is the predictive power of the average score across those tests. And so we're getting pretty high. This is like 36% of the variance up here, which is astounding for this sort of thing. And that's for the attention construct. It holds for the perception, for the declarative memory, language, cognitive control, and working memory constructs. So all of those predictive power goes up by averaging those behavioral scores of those 5 tasks. So this is using the brain data to tell us which of the behavioral measures are kind of relying on the same circuitry. We get a composite score from that, and we do better in terms of this predictive power. Now, sadly, if we look at the second cluster here, we do worse, and there's nothing there's no third cluster really. So this is looking at the second cluster. So we can't carry this very far. We might actually be able to look at maybe just doing these 2 things, and then, I don't know, there's some way to play around there. But if you have a bunch of behavioral measures, this might provide a smart way of averaging them to get a better handle on the brain behavior relationship. Although I would also back up and say it might muddy the interpretation a little bit, in the sense that you might be taking scores from disparate behavioral things and not really know what the component is in there that you're, it's going to be the attention component if the attention score is very high. But it can make it a little more complicated. So that's hierarchical clustering. So again, I want to get at this sort of the brain can feed information on the behavior, and the behavior is obviously needed for us to build these models and get information on the brain. So I think they go hand in hand, and I think there's a lot that we could do with this sort of technique. One of the things I'm not really going to talk about, well, I am going to talk about this some more. Another thing we can do is, I talked about these normative models. Are there any questions on that part? Okay. Well, I talked about, building normative models here and getting more samples and looking at outliers. So we actually had a paper where we looked at outliers here last year in Nature, and we showed that individuals for whom the test was failing, it turns out that there are cultural differences and things, and there's bias in the tests, and these biases are well known in the psycho and the whatever it's called, the PPI literature. But it's kind of new to us. But there's biases in these tests, and it turns out the brain is telling us when there's these biases in the tests, and that was kind of cool. But we can take that a little bit further and into the clinical applications. And what I want to do there is we can start to look at, well, who's fits the normative model and who doesn't fit the normative model. And once we account for biases, are there outliers that define patient groups? And so this is in this whole research domain criteria formalism. The idea is, rather than having categorical diagnoses, which is what psychiatry currently uses with the DSM-5 here in the U.S. or in Europe, they have this ICD-2 manual. And this, clinically, this doesn't work very well. They've done this for like 40 years. The groups are heterogeneous. They're not good at predicting patient response to treatment or predicting outcomes. So there's a search for an alternative approach and a more biologically based approach. Thomas Insel introduced this more than 10 years ago now, a search for a biologically based system where everybody's on some spectrum, and this is a dimensional approach to psychiatry. So before I was talking about how we all had attention scores, so every one of us is somewhere along the attention spectrum. Now we can talk about these clinical scores, so every one of us is somewhere along some paranoia spectrum, right? And other sort of measures like that. And so you could assign a paranoia score, let's say, to everybody, and then try to model that and look for the circuits and look for dysfunction in those circuits. So that's trying to use this approach then to put psychiatric illness in a more biological basis. The other assumption here that Thomas Insel introduced was that because psychiatric illness is a circuit disorder problem, we should see that manifest in looking at the circuits supporting cognitive function. So again, that's where they came up with those 6 cognitive constructs that I used. And the idea is that we should be able to see evidence for this in cognition. And what I'm going to show next is that, yes, but somebody can, what I hope to get at is that we may see people employing alternate circuits in order to achieve normal function on those scores. And so I think we have an approach now that can get at that sort of question, although I don't have the answer for you today. So again, this is now saying that the formal ways of diagnosing patients as schizophrenic or manic depressive or things like that are not that good. Let's put everybody into the same pool. And so we're doing a transdiagnostic sample, which is almost up to 300 subjects now. And everybody who has is on some paranoia spectrum, the circuitry for paranoia is going to be the same, but where you are, how your circuits are engaged is a function of your symptom score. So again, looking at these on a spectrum. So the RDoc introduced these different domains. We're really talking here about the cognitive domain, and within the cognitive domain, they had a panel of experts who defined these 6 cognitive constructs. And obviously there's lots more things you could put in here, and actually I'm interested in that question too, but this is what's defined in the RDoc formalism. So given that, we could apply these models to individuals and then see, do they form outliers? And actually we have kind of a complicated space here, right? So this is for one behavior and one brain condition, let's say rest or a task. But actually we have 16 behaviors, and so we have a model like this for all 16 behaviors, and we could start to do things like look for outliers. Maybe these 3 red dots, these are people, maybe these 3 people form a group. They're outliers on task 2, test 5, and test 7. And that's saying that maybe the model doesn't work for them, and why is that? And then if we look over here, the 3 blue dots are maybe individuals who form another group. And can we start to pull out, subpopulations here? And these are individuals who are somehow using different brain systems to, they may have normal performance. So if you look on the y-axis, that's their behavioral score. They may have normal performance on the behavioral tests, but they might be achieving that by using different circuits that aren't part of our normative models here. And so we're getting deep into this now, of trying to identify these individuals and maybe identify what compensating circuits they're using. And so I think it's a potentially powerful tool there. So there's a multidimensional space. We can assemble these groups, then maybe subgroups who require different brain behavior models and along these different dimensions. And so this is gets into a deep learning sort of problem, or a hierarchical clustering problem, or something like that. There's lots of ways you can look at that space, in other words. So we have a bunch of cognitive tests. We have some clinical measures. There's many subscores that we can these are the gross sort of categories here that we're looking at. But there's many subscores. But just a word here, the cognitive testing is generally pretty quantitative, right? You get a reaction time, you get an accuracy or something like that, and that's a quantitative number that you can stick into a model. The clinical tests tend to be descriptive, self-report, qualitative. There's no reaction time equivalent for depression, right? Or anxiety. I wish there was a measure like that we could attain, but there isn't. And psychiatrists, typically in a clinical environment, their clinical assessment is qualitative, right? They're not looking at reaction times. They're looking at the big picture, and they have a checklist of symptoms and things, but it's all either self-report or it's reported by the psychiatrist observing the individual. Now, if you ask the psychiatrist, that is the gold standard, and each individual psychiatrist does a really good job at that. The field as a whole fails at that, but any individual does well. So what we're hoping is to build models on these assessments, but actually we struggle. I was talking to John Gabrielli about this earlier. We struggle to build predictive models on behavioral or on measures like anxiety, and these are self-report measures. And so, and sometimes if you're talking about autism, let's say in kids, it's not even self-report by the kid, it's report by the parent, right? So we have many layers of filtering there. The idea with the whole idea of looking at the brain is that, even a behavioral test is a filter, right? So you have the subject's brain, you have them interpreting the test, and then you have you interpreting the test score. And so there's several layers of filtering there. And then if you look at autism for kids, you then have another layer, which is the parent interpreting the child's behavior. And so these things can be somewhat removed from the brain, and we often don't see nice, clear brain correlates, which maybe points to, a problem with those sorts of evaluations. Maybe not with the brain behavior modeling, because we can do it on things that reliably assess the brain without so much filtering. So there's this whole filtering, and the hope of brain behavior modeling is that we get rid of the tests, even the behavioral tests or the cognitive tests. We get rid of those and look directly at the brain, and then there's no filtering, right? We're looking directly at the brain. Still filtered, I guess, by our algorithms and by the bold signal, but less of this filtering by a test and things. So this is an open question. I don't, have an answer for how to build models on self-report measures. But it suggests that there isn't a clear link between brain and behavior for those measures. And maybe people are interpreting, pain, I think, is another one of those subjective ones, but maybe people are interpreting things differently. I was asked a question today, I think at lunch, what about other information? So there's many other types of information that people are starting to collect. You have Alexa listening to conversations constantly in your house, potentially. That could be a rich, source of data. You have lots of wearables. So everybody often wears a smartwatch. You get heart rate, you get temperature, you get motion, you get location. Smartphones and tablets. So EMA data is really, NIH just had an RFA in the spring about collecting ecological momentary assessment data. So, individuals have an app on their phone and it prompts them and says, where are you right now? Are you home or out? And they can say whether they're home, are you alone or with somebody? And you get measures of socialness, you get measures of activity, you get measures of sleep, awake. And so there's a lot of interest in trying to, incorporate that information. And then just, web surfing history or what they're doing. There's a whole, I stole this, and sadly, this was from some NIH symposium in the spring, and I took a screenshot, and I don't know whose this is. So if it's yours, I thank you very much. I don't know whose this is, but I stole it from a talk that I was in a Zoom meeting on the NIH. If it's yours, it's great figure, thank you. So there's all these scales of data that you can collect, and we're interested in particular, so I've been talking about cognitive assessments, exercise fitness, that's another thing, sleep is a big one. I think Russ Poldrack did a thing where he scanned himself, 149 times or something over the course of a year, and the primary thing that varied, was how much sleep he got. You can see that in the fMRI data. So sleep is a big factor. But all of these things, as we get more and more data, all of these things may contribute stress. And obviously hormones play a big role in brains and brain and behavior. And so, and then immunological markers, people, especially post-COVID, with long COVID, people are interested in this. So all of these things, we began with kind of brain and behavior. For brain, we could use more than just the fMRI. So the question was asked earlier, can you use, DTI and things like that? Sure, you can put that in there. You don't have to be limited to just this cognitive data. We can put in many different types of data. Every time you add these things, though, you need more subjects. So, and then on the brain side thing, we'd also break this up into different networks and stuff. And so these are kind of where we're going with this. But as we build these larger data sets, we end up with 10,000 subjects or something, and maybe the UK Biobank is providing some of that data. We need, then we can start to develop these sort of AI approaches to link these different elements and improve our understanding of all the different factors that are influencing the brain and the measurements that we get. We already know, coffee and sleep and sex are three things that are pretty major variables in our results. Sex differences are huge. Here I haven't done anything sex specific, but we already know these are things, and the more data we have, the more we can start to take into account these other factors, and maybe they can help inform the models. So I mentioned kind of briefly atlases. There's many atlases available. This is the Shen Atlas we have from our group, actually. This general approach is kind of insensitive to the atlas that you choose because we do that collapsing. So we basically, these edges are dependent on the atlas, but we basically sum something like 700 edges or 1,400 edges, and after you've done that, the effect of the atlas is pretty minimal. So the atlas isn't critical here, but there are some things to consider with the atlases. I'm trying to get a paper published right now on a consensus approach to atlasing. So if you don't know what the right atlas is, maybe you could define the atlas. What we did was 1,000 randomly generated atlases, and we did CPM, and then we found the overlapping voxels and made those into nodes. And so we're not dependent on which atlas we started with because we started with 1,000 random ones. That's an approach. Another approach is, so that's incorporating the behavioral measures into building the atlas. I just got a grant actually to kind of do the atlasing and the modeling in a joint sort of Bayesian framework, so you do it all at once. And we'll see how that goes. That's complicated. You could skip the atlas entirely, ICA approaches like Vince Calhoun does or Steve Smith. That only gives you a few components, but it seems to be a powerful approach if you know how to edit those components. And predictive power is about the same. They don't seem to outperform what we do. I've already talked about static versus dynamic. Oh, no, I haven't talked about this. I talked about tasks, but you could also, somebody today mentioned they're interested in dynamic connectivity, and certainly you could look at the dynamic properties of connectivity over time. We've used edges or z-scores here for the edges. You could apply graph theory measures here and put those into your model. I always get asked, so I thought when I was growing up in physics world that eloquence was using the simplest model that worked, right? So I have a simple model, y equals mx plus b, that works, and I constantly get reviewers saying, well, you're just using a simple model, why don't you use deep learning? But anyway, if you're that reviewer, the reason is that it's simple and it's easier to interpret. But we have since done things like ridge regression and nonlinear models and things. You take into account nonlinearities, which gives you a better fit at the risk maybe of overfitting, but it does generalize. So you can use whatever you want there as a model. I thought that it was eloquent to use y equals mx plus b. But you can put this into a deep learning framework, and a lot of people are working on this kind of stuff now. So we've got circuit identification, component contributions, testing the tests, like what are the tests actually measuring? Who are the outliers on those tests? Are there biases in those tests? We want to apply this in an individualized medicine approach and group these models to identify individuals that might be part of a different group. And so that's this whole brain behavior phenotyping across many tasks. And so ultimately, I think the future is bright. We're just at the beginning of all this stuff, and I think that there's a lot to learn here. We're going to understand a little bit more about the brain circuits that support behaviors. We're going to look at sub-circuits or subcomponents of those circuits. We're going to develop normative models for those circuits and then identify subtypes from that. Or I think it's particularly exciting to maybe look at compensating strategies. So what people are engaging different brain areas in order to achieve normal performance on a task. And we'll learn more about confounds as well. But as more data is integrated, these factors influencing these relationships are going to be revealed, and hopefully we'll get better models. So thank you very much. A ton of people do this. And happy to answer questions. Yeah.
Amazing, thanks. Really a tour de force. I see why it's 400 pages.
It's not 400. Anyway, thank you. That's an exaggeration.
As you know, as a grad student for a long time, I'm curious if you visualized any effects related to expertise or training, and I'll parameterize that just what I'm wondering about outsizing. You throw, let's say, 150 in your sample at an hand-back task, which is not picking up data bases. You get those 1,400 edges that relate somehow. It's relatively complex. Say it's only 2%. Train one group, don't train the other. You spend a lot of time doing handbacks. They do the same thing. What would you see? Have you done it? What would you hypothesize? Maybe you get 800 edges, 400 edges. They're relevant. Would you get less variability within the sample? So you have to talk much about what does that sample look like?
Yeah.
You take these 100 people, these 10 people, do they have similar edges activated? These are parts of the brain that are actual behaviors, variability. There's a lot of variability in strategy behaviors for the first time. Would you reduce some of that variability in strategies? Interrogate strategies and plus that.
Yeah, I think that's a great question, and the first answer is no, we haven't done that, but that's on our list of things to look at. And what I think my hypothesis would be that, so we have a set of edges, let's say 1,400 edges, and those edges are changing with performance, right, across people. And so if you're down here at the low end, I think your edges should look more like the people up here at this end as you train and get better. You should move along that curve, and you should end up being up here.
Yeah.
And we could potentially track where you're going by looking at that set of edges and seeing where you are on that curve. Now, having said that,
it's not just about which edge. It's like we're trying to understand about the brain in reverse, right?
Yeah.
What would be the atlas? What would be the right circuits here? So if you can get rid of the noise from big hard task and get to this particular type of task with this strategy, then that might be a little more informative about what the brain is doing in those.
Yeah, so the other, I was going to say the other thing is that not only, so one scenario is that you move closer to the people that are on the high end of the spectrum, right, performance spectrum. so your edges look more like them. The other is that you employ, for some reason you've never gotten there, and now with training you're going to like employ some other mechanisms, and so maybe you bring in other areas and you overemphasize some circuitry in order to perform better, even though you didn't learn originally, you know, the proper circuits. So those are two different scenarios, and I don't know. That's a great question, and I think we're very interested in training. I talked with John a little bit about, you know, one of the things we'd like to do is predict outcomes, right? Could you predict outcomes from learning and is it dyslexia in a reading intervention, right? Do you start to look more like the good readers? And I don't know the answer to that, but I think that's a great question, and that's something you can do with this sort of approach. Yeah.
Similar to that, have you looked at building models based on treatment response? Because you were mentioning that there was a lot of, it was challenging to use a psychiatrist's analysis as an input or something to model.
Yeah.
But if you look at treatment response, you at least can get, you might be able to get a little bit of actual quantitative data.
I would love to do that. I don't have the data to do that right now. I don't have treatment response, you know, controlled trial outcomes with fMRI. If you know, that's where we're going, and ultimately that's what we need, right? Like, what's the fundamental measure of success here? The fundamental measure of success is if we can either predict treatment outcome or, you know, hopefully assign people to the right treatment arm. But we don't have data on either of those at this point. So, but that's a great question. Yeah. Behind, yeah.
Okay, so I have two questions. One follow up with that one. So you don't have the trait or treatment, but you know, each healthy brain network, that data set, they have the eye tracking data.
Yeah.
So maybe that's kind of the behavioral data, and then it's that's filtered by, you know, their self-report, parents' report, or something like that, and they also have the brain part. They have the EEG part and the fMRI part.
Yeah, so the eye tracking, we've done a little bit of that at Yale with autism, kids with autism, and I'll tell you two things about that experiment. One is that the eye tracking is great, and you can, see whether they're looking and why, but you can also get arousal, right? I don't know how controversial that is, but you know, certainly in mice they use eye tracking to look at arousal, or pupillometry to look at arousal, and you can do the same thing in humans, although that's more controversial. But I think, you know, you can put that in as a factor in the model, and you can do multi-dimensional models with many factors, so by all means do that. One of the things I was going to say about the Yale study in autistic children is that we were imaging kids, I think 7 or 8 and up, and we had a task. It was a slowed down, easy version of the grad CPT attention task, and we actually had more successful experiments with kids doing that task in the magnet than we did just doing rest. Like, just having them engaged in something made the experiment go faster, and they stayed in the magnet and moved less. So I would encourage, again, you know, trying to collect connectivity data under task conditions, because that was a case. Actually, I had that paper in Scientific Reports, I think. Better outcomes just in terms of these difficult populations that are difficult to image. Does that answer? Did you have a second?
Actually, that's not my main question.
Oh, okay.
So my second question was, sorry. I remember, so there was a table, each column was like 6 cognitive tests.
Yeah.
And then the rows are those things they did in the fMRI scanner, right?
Yeah.
And then the best predictor, not predictor, anyway. So movies and emotion eyes.
Yeah.
And then you were saying, if it's just synchrony, right? And then they should, it should be one thing that.
Yeah, one row would be the
yes,
best.
But that's not the case. So I was just thinking, especially for the movie part, naturalistic data. But each movie they would be so because they are naturalistic, so they have some unique aspects, each of them. I just want to know for different type of movies, like have you ever tried something like break them down, take the movie?
No, we talked about that. So, you know, you can do state induction, right, with different movies. So we talked about doing, you know, CNN clips of disasters, and then some happy thing and stuff. The movies we have here, so I've been a subject 50 times, so I know this very well. But the movies we have here, it's The Princess Bride, part of it's like 2 minutes of The Princess Bride, 2 minutes of Up and Away, kind of a cartoon thing, and 2 minutes of Inside Out. And so these are kind of kids animated things, Do you want me to recite them? No. But anyway, so, we're not really inducing mood changes or things like that, but certainly people have talked about that. I know Simon Eckhoff in Germany has talked about doing different clips. Actually, he was collaborating with Emily Finn to do that. But yeah, that's I think could be a very rich area to explore. So one of the things that's funny about this, I got into resting state fMRI because I was like, I don't know what to do with task stuff anymore. I'm not going to do 101 iterations of a task. Okay? Just don't know what to do there. And resting state fMRI seemed cool. And now I'm back to tasks. So I don't know. There's a question there,
I guess I'm wondering, do you think of this is approaching the notion of, is there an ideally organized brain? Like if we find that people who are more dissimilar from the Atlas connectivity, or the Atlas pattern, which is probably developed off of a group of healthy participants, do you think that mean that there's like some kind of notion of an ideally organized brain?
Yeah, that's a good question. I was working under the notion of finding the ideal brain state for looking at the brain. But now you're saying, is there an ideal brain? There probably is an ideal brain, obviously, for each of the different measures. I don't know if any one person, we haven't found that one person that is ideal on all the measures. But yeah, I'm sure there is an ideal sort of, you're wired just perfectly in just the right way that you outperform everybody on every test. Probably, obviously we have people that are at the high end of the spectrum on some of these. Sadly, if you look, I do have my data on these things. I'm not at the high end on these measures. But that's an interesting question. I was like, so, we can manipulate brain state, and here, let's say these are manipulating brain state. I was kind of thinking, is there an ideal brain state that puts you in a framework where we can really, get rid of all the extraneous stuff and we see, what's important? The answer is it's complicated. Yeah.
The sense in the field for a long time now seems to have been that connectivity is what's most interesting about the resting state. How true is that? Is there some way that you can find answers to it in your data?
What do you mean most interesting about the resting state? I've never heard that.
People don't look at power spectra.
Oh yeah, well there's little, yeah, okay, well, so the power spectrum is not very interesting because bold is a slow thing and it's filtered, it's heavily filtered, right? It's a really low frequency, like you can look at the canonical response, take the Fourier transform of that, look at the power spectrum, but it's not very interesting. It's got a bunch of low frequency information, right?
It's not interesting for someone who does EEG. But
well,
nevertheless, it is the power that's driving the connectivity as we measure it in fMRI.
Yeah, but I haven't seen anything interesting from people that have looked at that, I guess, let me put it that way. People have looked at that, and I don't think there's much there. And I think there's not much there because it's just really low frequency.
Their amplitudes also, you heard it, I guess it was
yeah,
brought up
the
in the question
amplitude
there
stuff,
about
you mean the
amplitude.
amplitude of the fluctuations?
Exactly.
Yes, and that also is, that's heavily sensitive to caffeine and stuff like that. Vasodilators, drugs and stuff like that, So yeah, I don't also, and obviously correlation is sensitive to, amplitude falls out of correlation, but signal to noise doesn't, right? And so you do tend to, with higher amplitudes, you do have higher signal to noise, you get slightly higher correlations. So, this stuff is somewhat sensitive to amplitude.
A mouse person, if I just push back on this a little bit, a mouse person who wanted to know, well, where in the brain is X happening, where X is a behavior. The first thing that they're gonna do in many cases is they're gonna look at this CFOS marker, right, early immediate gene activation. It's, who knows what its relationship is to anything. It's definitely not fast. And it's got nothing to do with connectivity, and yet it is something that's fairly widely used in the field, with plenty of controversy around it.
Well, part of the problem with bold too is, right, it's plumbing. So it's a complicated response. I think, again, in humans, people have tried to look at some of this stuff, and people have tried to look at fast responses. I think the Bandettini group was trying to look at the upslope of the bold. I haven't seen anything that like really caught on, or there's an initial dip, right, as the oxygen consumption
We'll
hits.
talk about that.
Yeah, okay. So I don't know, I mean.
Okay, so connectivity's it.
Yeah, connectivity's it, and all these other things are like too complicated. Okay. Yeah.
Yeah, I was just wondering how much data do you actually need to build reliable predictive models? Because we actually have data from a sample of kids with ADD, about 10 minutes of the grad CPT. So I was just curious whether that would be feasible, and also was wondering, maybe with certain samples, if you have like really reliable brain activity, low variability, you might be able to predict better with like smaller amount of data, whereas in other clinical samples.
Well, that was going to be my answer to your first part, which is how much data you need. I mean, it depends on what if you're doing grad CPT, you're interested in attention. Attention typically needs less data. So 5 minutes is probably not enough, 10 is better, 40 is ideal. And then, how many subjects? I mean, Monica Rosenberg had success with 25, Yale undergraduates. But that depends on what your subject pool is. Are they kids that throw around a lot? Are they, good subjects? So it depends. The answer is it depends. You saw from this that different behavioral measures have different predictive power, and so it depends on that too. Looking at your health areas. But 10 minutes of data, you might be able to see something like that. It also depends on your imaging parameters and things like that. Yep.
It's an interesting talk. So you mentioned about the anxiety. What data do you have? Why?
About what? Sorry?
Anxiety.
Anxiety. Yeah, so to be honest, I'm sorry, I can't quote what measures. So this is Dylan Gee's work. She's published a ton of papers on, activation maps and anxiety, behavioral studies and anxiety in kids. I can't tell you what the measures are that we looked at. We looked at 3 or 4 different measures, and in 2 or 3 or 4 different populations with up to 150 subjects in some of the groups. And we really didn't see anything that was reliable, like that we could predict reliably. Yep.
I was struck by your motivation for the talk for the basic science part, where you say you want to build predictive models of behavior to figure out what parts of the brain are involved in a task. And it's different from, I guess, the majority of the field, because oftentimes we try to figure out what parts behave similarly across people, right? We'll look at the group responses, kind of what are the commonalities. And you're saying that you want to see what parts predict individual differences to identify the basic components of behavior. Do you think these two approaches will ultimately lead us to the same answers, or are they complementary? Do they produce kind of complementary?
Yeah, no, that's a really good question. So we do see, they're not the same. I do think they're complementary. And I think what you're pointing out is really important. And I kind of mentioned it at the beginning, like where there may be circuits here that are involved, but not relevant, right? And remember, this data is not collected when they're doing the task. This data is independent of the behavioral test, right? So there may be circuitry in the brain that you do need to do the test that we're not seeing, but it's not differentially involved. And it's almost like, in fMRI, there may be lots of brain areas, and this would suggest there are, lots of brain areas that are involved in the task, but they're not differentially involved between the task and control. And so you don't see them, or they're just below the threshold, cutoff threshold. And that's a problem with fMRI and neurosurgery, right? It's like, you can see actually it's the opposite problem there. You see what's involved, but you don't see what's critical. And so, there are these subtle differences, but that's a good point you raised. Yeah, we're looking at what's differentially involved as a trait, not what's differentially involved at the moment, because we're not doing this simultaneous with the behavioral measure.
I'm gonna,
One
that's
more.
good. Go for it.
Great talk. I was wondering, do you kind of see variance in functional connectivity along a kind of sensory fugal organizational axis, with higher variance for more kind of higher order cognitive functions, lower variance for more like basic sensory functions, and maybe that could explain part of the predictive performance?
Yeah, there's definitely that. I would agree that that is the case. Whether we see that or not, we haven't really noticed that. When we look at so we can break up these networks. So we can do CPM on the whole matrix, and then we can look at, well, what networks. We can take the Yo7 or the Yo17 networks and say, are most of these edges in one network or the other? And to be honest, like for IQ, it's frontal parietal and then some temporal, but for the most part, there's always contributions from almost all the different networks, and so we haven't focused on that a lot, but there may be some relationship there between variance and also those networks are more. The sensory motor systems are much more well defined and easily overlapping across subjects, whereas some of the cognitive stuff is not.
We should now transition to the reception. Thank you.
Well, that's a whole nother talk. There's a lot of detail under there that we're ignoring. But I think it's a good question, and not enough people ask that question, actually. That's one of my pet peeves.
Yes, be worrying about that.
Yeah, there's a lot underneath there.
Including, astonishingly, people like Frank Tong, for instance, have done decoding of pixels or voxels, and I'm astounded that works because under each pixel is 10,000 neurons. So different sorts. Some of it going opposite direction, excitation and inhibition. And I study those circuits. So I am always stunned that you can do this.
Nancy Kammer sure does MVP. It works.
It probably disappears from a lot of the MRI.
It is possible. Yeah. So
No,
that related to the question.
There's a lot there, and I do think that the brain is flexibly organized. We do kind of have the assumption that it's pixel, everything's doing the same thing, but you're right, there's 100,000 neurons, they're not all
Never
doing
make
the
that
same
assumption.
thing.
And also your point about that lamppost that we used to.
Where we look, yeah.
That's where we look.
Yeah.
And so we expect something that looks like an atlas, and it's basically about a phonology.
Yeah.
Doesn't make sense.
But since we have made you work so hard, please come and have some wine.
Yeah, sure.
I know you are in good hands with them and dinner and all that, so thank you.
Thank you.
How are you?
I'm good. How are you guys?
I am thinking about it now, because your work is really cool, and I was wondering if your lab or other labs that